What is biotechnology? Well, it's pretty much just using technology based on biology. It's as simple as that. It is the power to harness cellular and biomolecular processes and technologies to develop products that improve biological health. It allows us to combat diseases, find cleaner alternatives, and most importantly, prevent biocatastrophes. Now, in this video, we'll specifically be talking about cloning. Now, against popular belief, cloning isn't a very quick process where you put in a guy and 10 minutes later a new guy comes out. No, it's completely different. It takes a lot more time to create this embryo, and it grows just like a normal organism. Cloning is just taking a bunch of cells, creating an identical embryo, and growing it as normal offspring inside a parent body.
So, let's see what they used to think about cells. They thought that differentiated cells would only stay the type of cell that they are. So, a skin cell could only become another skin cell. But embryonic cells could be whatever they want to be. It was a whole different story. Embryonic cells could be a heart cell, or a liver cell, or a lung cell, whatever it wanted to be. It was its choice. That was the main difference. As a matter of fact, if they took an embryo and they placed it inside another organism, what would happen is that they would get the same organism back. So they tried that with a frog and it actually worked. So now, let's take a more focused example like Dolly the sheep. At Roslin Institute, a place in Edinburgh, University of Edinburgh actually, they took a sheep. Now what they did with the sheep is they extracted a cell from it. So the cell was the egg cell from the sheep. After extracting the cell, they took the nucleus and they took it out of it. So now we have an empty egg cell. They took another sheep, and from this sheep, they took out another cell. Just a random cell like a skin cell or whatever. And here is a random skin cell. Now from this random skin cell, they put it into starvation conditions. So what happens is this random skin cell under starvation condition acts now like an embryonic cell. Now, this embryonic cell, when it's used with a very small tube and electricity, the nucleus transfers from the embryonic cell to the empty egg cell that we had from earlier. So that's their transfer. And now we have an empty cell, egg cell, which divides just like an embryo. This embryo could be placed back into the sheep and it would make more sheep just like it. Like that small sheep right there.
Okay, so let's take a look at why this cloning actually worked. So, since all the genetic information is stored in the DNA, it means that each and every single cell in your body actually has all the genetic information you need. So, one cell in your body, say a skin cell, has all the information that a brain cell, a muscle cell, a blood cell, every single cell in your body would need. But it only uses whatever part of that information it wants, so whatever skins need. So, now by putting all of this DNA from the nucleus into a new cell, this DNA will control this cell. Now this DNA is acting embryonic because of the starvation period that it went through. So now this embryonic cell, the new cell that we got, is going to create an embryo pretty much. So it's going to divide and create an embryo. This embryo can be placed inside a parent organism, and then the new organism will be pretty much just what you copied from, where you got the DNA from. So those two will be exactly the similar. But the organism, the embryo that we place, will grow just as fast as a normal child would grow.
Okay, so we can look at many advantages of cloning. One of them includes the benefits to modern medicine. We can be able to treat degenerative diseases because cells mimic other cells. We'll be able to treat diseases like cancer and Alzheimer, replacing the bad cells with the good ones. We can also make new organs, like pig livers have been successfully transplanted into humans. So if we can clone pigs, we can have more livers that can be transplanted into humans. And this could also go for a lot of other organs, so that there is no long waiting lines, so less people die. The second one is we can help infertile couples. If you can't conceive, you can probably get the embryo placed in you, which means that you can have a child of your own. And the best part is that you kind of have a choice on what you want in the child, right? And some people can look at it in a good way, some people look at it in a bad way. It's your choice. But sadly, it's banned because of human cloning, because of some ethics. But we're struggling with that rules right now, and possibly this is a good advantage. The other thing is that we can reverse the aging process. Old people, what they need is new cells, and cloning provides new cells. So if we can put the old cells in the old people, perhaps we can perhaps we can lengthen their life, or maybe even make them look a little bit younger due to cosmetics and all that. We can also protect endangered species. The thing is that more species are dying out. So what we do is, just like we created Dolly, artificial sheep, we can create artificial species, artificial animals of the endangered species, so that they don't die out when they need to. They'll keep living on forever. Also, we can improve food supply. Now, hunger is a main kind of topic in the world, how we can feed world hunger. This way, if we create more meaty animals such as cows, pigs, or whatever, then we can feed these extra people who are still hungry in the world. And perhaps this is a good alternative and a good solution to that main world hunger kind of problem. So now let's look at some disadvantages. The first one is uncertainty. As you remember, Dolly the sheep, 1 out of 277 actual embryos, Dolly was the only one that was there. That means that 276 of them died out. And even if we get a number that is much more suitable for us, we still have that uncertainty as if they're gonna die out or are they actually gonna live. And is this kind of right? Since embryos are now considered life, are we actually killing it? The second thing is inheriting diseases. It's true that we get more cells when we get cloning, but the truth is that there are a lot of diseases hidden in the cell itself, such as some genetic diseases that are because of mutations in the DNA. Now, if the cell were to regenerate and create a whole new organism, that means that each of these cells would have that genetic disease. That means that the new organism would be inheriting the disease and spreading it. The third thing is potential for abuse. We know that movie stars like to use their money for bad stuff. And technically, this was only made so that we can help medical research and help those people who actually need help with their health. But is it right for actual movie stars and big shot popular people to use this and make themselves look younger or make themselves look more attractive? Anti-evolution. Darwin said that the survival of the fittest, the strongest will survive. But now we're kind of choosing who survives and who doesn't. So we're kind of going against evolution, and this is all artificial selection now. That means that if we choose who survives and who doesn't, a lot of change is going to happen in the world itself. So we don't know the effect of that. There could be some adverse effects, and that also goes back to uncertainty. What will happen if we actually do it? And ethics, finally ethics. Is it right for us to choose to make new organisms, to make new life, or is that actually God's role? Some people look at it in a religious way. Is it right for us to choose that this person can have a new himself, like a clone of himself?
So what's my opinion on all of this technology? Well, I believe that is in fact great. By using cloning, we can cure a lot of degenerative diseases like cancer and Alzheimer's. We can also harvest organs for organ transplants. So we can take a pig liver which has already been transplanted into humans, and we can make more pigs with more pig livers so that the lines for organ transplants would be much shorter. Now, suppose that we were about to take a little bit more humanitarian approach to this situation. Well, we can see that there's a lot of advantages in that also. We can see that this cloning technology will create a lot more food, because we can clone pigs and cows and everything else that we can eat, and we can definitely end world hunger with this. Yes, chances are that at beginnings we'll probably end up killing a lot of embryos, but I believe that with right research and right commitment, we can see a lot more potential uses for this technology. Now, however, in Canada, we have a lot of laws that prevent us from doing some kind of wrongdoings with cloning. So, the Assisted Human Reproduction Act prevents the creation of human embryos or in vitros. This means that we can't have chimeras and hybrids, so we can't have like a half horse, half man kind of thing creature. Anyways, who deserves kudos for this technology? I believe this man does. Yes, this is Oswald Avery. He pointed out that DNA is actually the genetic material. He kind of used some of the research that Frederick Griffiths did, but I guess he deserves more of the credit. So if he didn't actually point out that DNA had all the genetic information, we'd be looking all through proteins, because that's where we used to think that the genetic information was. I know, crazy, right?